Hi guys, we're back again. Week 5 of isolation. So week 5, week 6, we're going to actually look at studying ecosystems and get you outside and unfortunately into your backyard. Well, not so unfortunately, but not our reserve at school or a natural ecosystem, just one in your backyard. If you've got a local reserve close by that your parents are happy for you to wander into, you probably could. But I mean, obviously it's easy enough to do in your own backyards too. So what do we do when we're investigating an ecosystem? Well, we need to get information about the abiotic and biotic factors. And there's three things we're sort of keen to know about all those factors. One's distribution. So, where do you find it? I can't remember what I dragged out here, some distribution map of some plant, which is interesting because it's growing in quite strange diversity of locations when you think about it. But they're all coastal, so clearly a coastal plant, but also found in Papua New Guinea. So there's something's influencing this plant. Why does it grow in these locations? They're probably wetter, but yeah, anyway. So we're looking for that sort of connection between the abiotic and the biotic, and here's where you find this particular organism. We also want to know, is it abundant? So do we see a change in abundance? So there's a lovely graph you could quite easily make from quadrat sampling. This one's sort of over time, so obviously watching a few species over time, but this one's obviously most abundant. Certainly most abundant here in May. Let's say this one falls away completely in September. So we're seeing change across the seasons, so in relationship to abiotic factors, water, food, heat, and also this one's obviously a dominant species in the area. These ones in less amount. So looking for that sort of information, we want to know, are they abundant? Are they not abundant? Why might that be the case? So they're abundant at different times. So we're looking at migratory patterns. And we want to know about diversity. Just how many plants and animals do we have? Again, does it change throughout the year? Does it change through place to place? The idea of distribution of that diversity is really important. It gives an indication of animals which live within certain plants, so there's certain associations due to habitat. And that's some information we want to try and find out when we're out and about. That's a lovely little quoll there. So how do we do that? Well, we use things called transects, which are basically straight lines, so a straight line across your environment, and then you set up quadrats regularly spaced along that transect, and you take information from the quadrat. And when we're looking at the quadrat, I tend to use a thing called the Braun-Blanquet system, which I was taught when I was at university all those years ago. It probably changed now, but it works fine. And they probably use similar systems, but Braun-Blanquet was a big thing in the 80s, and Braun and Blanquet are two Belgian scientists who developed this process. So, how's it done? Well, there's a lovely one I would have done in the reserve with you. We have our transect, that straight line. So the blue line here is our straight line, our transect. Normally set up along a compass bearing. And these red blocks form my quadrats, so four-sided figures. There's a mathematical formula to work out how big your quadrat should be given the tree types and plant types in your environment, often taken by sampling the girth and height of the dominant species from memory. But if we're in the reserve, we would have done 10 metres squares, nice sampling size. In your backyard, you can just use 1 metre squares. Otherwise, it could be the whole backyard in one quadrat, depending on how big your place is. So to try and randomise things, you would start on one side of your transect at the bottom. That's a 10 metre square. Find out what plants and animals in that 10 metre square. Walk 10 metres, do another one. Walk 10 metres, and you're swapping sides. It's quite important to do that because it's easy to say, oh, there's that rare endangered plant there. I'm going to swap sides. And you're sort of skewing your sampling. If that's the only one that's in the whole area, then that's not very abundant. It's nice to note for diversity, but it also shows us that it's not actually that common. So you've got to be a little bit careful when you don't want to skew your data. Often, people will stand with a stick with their back to the transect and throw the stick, and wherever it lands becomes the first one, and from then on they randomise from there. Other people do complete belt transects, so every transect runs into the other. That's okay in a small space. In a large environment, and when I used to do this professionally, you're walking through the Alpine National Park, and a transect could be kilometres long. You don't want to belt transect. You want to randomly sample rather than take the whole thing. And then when we first started back in the lab, we'd spend days analysing it all and turning it into maps. So you get these lovely vegetation maps based on what you've taken and using the aerial photographs of the space to line up things. And so you'd be extenuating the lines. So you're going to do the same thing in your backyard, but not on a transect unless you go a particularly large backyard. So this Braun-Blanquet method is just a way of estimating cover. So it doesn't count the number of individual species of a plant. It just gives a feel for how much they cover your quadrat. So I've deliberately put these pink ones here, this big tree of some sort. That's not actually covering that much of the quadrat. And that one, again, not all the plant is involved. So you've got to take that sort of how much they overlay your quadrat into account. You've got to look up and down. That's not just one direction. So if there's less than 1% cover of a particular plant, we give them a plus. Then 2 to 5% gets a 1, 6 to 25% gets a 2, 25 to 50 gets a 3, 51 to 75 we call 4, and 76 to 100 gets called 5. So you don't need an exact percentage cover. It's a rough idea. Obviously, this is interesting because this is, in his case here, these are 2 trees, but they're giving us lots of cover. So it can skew a little bit. It gives a sense of that being really abundant when it isn't particularly. But the abundance becomes important when you start comparing site to site, so quadrat to quadrat. So essentially, what you need to be able to do is identify your plants. Now, plants are pretty straightforward in the sense that they're sitting still and you can take photographs. And if I was working in the field, we'd actually take samples of plants we didn't know. We'd actually know a lot of plants before we got there. But anything new, we would sample. We'd take flowers if we had them. We'd take fruits if we get them, leaves, stems. If there was very few of them, then lots of photographs and drawings. In those days, you wasn't any digital cameras. You get your film camera out, but you'd also do lots of drawing. You want to get those details, because those details allow you to key out and work out what that plant's name is. And often, of course, working in some of these locations, you'd find plants that no one recorded before. And so there were new plants to be recorded. And people got to put their names on things. I was just a grunt. I've got to collect data and help. So we can work out the plants, the diversity. We've got a list of plants. Animals are harder. Animals don't tend to sit still, especially when they see humans. They don't tend to trust humans very much. Some do. Some brown bandicoots are too bloody trusting for their own good. Nice little animals. So we tend to collect evidence they've been around. So the obvious evidence is poo, or scats as we prefer to call them. And we can collect scats and identify them. Each animal tends to have a particular way of rifling their poo, and they get different shapes and sizes and contents. Yep. Okay for herbivores because it's only plant material, but when you start getting the stinky carnivore poo, you really want a pair of gloves on, and preferably don't bother touching them in your little study because I don't want you getting sick. We can use our ears, of course, because lots of animals make noises for us, so we can actually, frogs and birds are quite distinctive in their calls, so we can actually work out what animals are there by what they're calling. Obviously, we use our eyes to collect scat information. We'll see runways through grasses, lots of burrows, nests, fine feathers, all that sort of stuff. And your nose can be quite good too, because especially the predators tend to mark their territories with a pretty strong urine. Foxes are well known for leaving a very strong scent behind, as are cats. So you know when they're around because you can smell them. And then, of course, you've got your abiotic factors to collect as well. Now, obviously, would have gone to our school stream, which is pumping at the moment, all that rain we've had, and got some water samples. We would have taken the soil samples. We would have done some pH testing and other chemical testing of those soils and water, working out what sort of stuff we've got present. That probably isn't that easy for you guys to do because, of course, you probably haven't got a stream running through your backyard. Some of you might. Some might have big backyards. One of the other things you can do, of course, is go to the Bureau of Met because they have all sort of rainfall, wind, and sun type data you might need. Take a little bit of digging, but you'll find that they're on the Met. And then you can collect your abiotic stuff in those other ways. So we'll spend one class going through this material, making sure you've got how to go about doing it under your belts. And I'm going to let you loose in your backyard to go get some data and prepare a report on the distribution, abundance and diversity of the biotic and abiotic factors of your backyard. I'll see you in class soon.